Thanks everyone for coming. We're starting a little bit late today. I know there were formal tickets on sale, but glad everyone's here now. Because I want to introduce Dr. Julian Lui, who is here from NICHD, from the section on growth and development. He's a staff scientist, has been there for the past 11 years. Dr. Lui is originally from Hong Kong, where he did his doctorate in biochemistry. So he can talk to us a lot more about mechanisms. I know a lot of us here are epidemiologists and public health scientists, but it's very important for us to understand those mechanisms. So his lab down in Bethesda is focused on a couple of different things. So some work on what controls body size and kind of when does growth stop, but also more translational and clinical aspects of kind of growth faltering or genetic disorders related to bone growth. So he's going to talk to us today about nature and nurture, genetic and nutritional regulation of skeletal growth. Can you turn on?
Yeah, I think so. All right. Thank you, Amanda, for the introduction. Actually, I'd also like to thank Dr. Keith West for inviting me to be here today. And my talk today, it's on genetic and nutritional regulation of skeletal growth. And you might be thinking, why do I need to learn about skeletal growth? I want you to think about this as more of an overall effort to try to solve childhood stunting in the world. So child stunting remains as one of the most significant impediments to human development. Globally, it affects more than 150 million children under the age of 5. Child stunting is associated with numerous long-term consequences, like increased mortality, cognitive deficit, decreased economic achievement, as well as short stature in adulthood. Now, in the past decade or so, there's a global call to reduce childhood stunting, and there's a particular focus, actually more recently, on the first 1,000 days of life. But often missing in this effort is an emphasis on the understanding of the biology of skeletal growth. Now, I believe that a better understanding of the process of linear growth is going to, among clinicians, dietitians, as well as public health specialists, is going to benefit tremendously in terms of strategizing how to improve and how to solve childhood stunting, and which is why I'm here today to talk to you all about skeletal growth. So so my talk is going to be divided into 3 main parts. First, I'll talk about, let's see how this pointer works. Okay, so first I'll talk about the physiology of childhood bone growth. I'll focus on a structure called the growth plate, which you will get to know a lot more after my talk today if you're not already familiar with it. Then I'll talk about the different factors that contribute to the regulation of bone growth, including different genetic factors and the environmental factor. I'll emphasize on how nutritional status plays a role in regulating all these factors. And then I'll move on to talk about the timing and duration of bone growth. How is that regulated? I'll introduce a concept called growth plate senescence, and I'll tell you how growth plate senescence is involved in a process called catch-up growth, which happens after malnutrition gets corrected. So let's get started. We know that children grow taller because their bones grow longer. And bone elongation is driven by a process called endochondral ossification. Now, the word endochondral ossification itself implies that the bones were being made, or this process is occurring inside a cartilage. Now, in our body, endochondral ossification happens in a structure, in a cartilaginous structure that was finally the ends of our long bone. That's called the growth plate. So let's first take a look at what is inside the growth plate and how does it work to drive bone elongation in children. The cells inside the growth plate, we call them chondrocytes. All these are chondrocytes. And structurally, the growth plate is divided by the histology into three distinct areas that we name them resting zone, proliferative zone, and the hypertrophic zone. Chondrocyte in each of these zones looks different. They also perform a different function, but altogether, chondrocyte in these three zones support childhood bone growth. Closest to the epiphysis, which is the end of the long bone, you'll find the resting zone, which contains progenitor cells. You can think of them also as stem cells that are capable of self-renewing and also give rise to new clones of proliferative chondrocyte right underneath. We know this because we have done an experiment in rabbits where we try to cut away the rest of the growth plate, leaving only the resting zone. And we found that the resting zone alone is enough to regenerate the rest of the growth plate. As these resting chondrocytes move down into the proliferative zone, they now become arranged into columns that are parallel to the long axis of the bone. And within each column, these cells divide rapidly. And as they divide, that also pushes themselves gradually down the column, almost towards the center of the bone. And as they gradually move down the column, as they get near the bottom, they stop dividing and start undergoing hypertrophy, which means that the cell become larger and larger. And so we call this area the hypertrophic zone. Now this process of chondrocyte stem cell recruitment followed by proliferation followed by hypertrophy also lead to formation of new cartilage tissue. That's because one of the functions of chondrocyte is also to produce cartilage matrix protein that makes up the cartilage tissue in the growth plate. So you might think, as this process goes on, what happens is the growth plate continues to expand, and this piece of cartilage would become thicker and thicker. But that actually doesn't happen. That's because simultaneously, at the bottom of the growth plate, there's continuous invasion of blood vessels and bone cells, which are also called osteoblasts. So these osteoblasts goes from the bottom of the growth plate. They produce enzymes that digest away the cartilage matrix protein and use it as a scaffold to start laying down bone matrix protein. Alternatively, there are also evidence suggesting the terminal hypertrophic chondrocyte, that is the last hypertrophic chondrocyte in the growth plate, can also directly transdifferentiate into osteoblasts. But in either case, that means as chondrocytes continue to proliferate and hypertrophy, there's new bone being progressively created at the bottom of the growth plate using the cartilage as a building block. And that's what makes the bone longer and longer in the child. And so from a simplistic point of view, all genetic and environmental factors, which includes nutrition, control or affect a child's bone growth ultimately by exerting their effect on the growth plate chondrocyte, either by regulating the stem cell recruitment, controlling proliferation rate, or affect the chondrocyte hypertrophy. And by and large, this has proven to be true thanks to some remarkable advancement in human genetics as well as genome sequencing in recent years. Back in a few years ago, there was a genome-wide association study, which is also called GWAS. In this GWAS study, they try to look for variants in the human genome. These variants are usually in the form of SNPs, which is also called single nucleotide polymorphisms. So these are places in the human genome, and they try to look for these variation and try to associate with the variation in adult height, which means trying to find places in the human genome between different people that would help explain why some people end up being tall and some people end up being short. And what they found is this GWAS study identified hundreds of loci in the genome, which means places or location in human genome that is associated with normal adult height variation. And many of these loci are located within or close to genes that are expressed specifically in the growth plate, or they have important function in regulating the growth plate. Therefore, confirming that the growth plate is really fundamental in controlling bone growth and controlling your final stature. Now, what I find more interesting is that many of these genes that regulate the growth plate depend on what kind of mutation or variant you have in that gene. You can produce a broad whole spectrum of phenotype involving childhood bone growth. So for example, mutation of one growth plate gene, if it's a homozygous complete loss of function mutation, could give you skeletal dysplasia, which is one of the most severe forms of growth disorder. Whereas heterozygous loss of function mutation of that same gene might give you syndromic short stature, which previously was called idiopathic short stature because they didn't find the cause of those. Mild polymorphism, like a single base substitution of the same gene, makes you a little bit tall or contribute to a person being a little bit tall or short. Whereas gain of function mutation can give you tall stature. And that's what I'm going to talk to you about next. So what are these genetic factors that contribute to the regulation of bone growth rate, and how does nutrition play a role in regulating all these different factors? So growth plate chondrocytes are regulated at multiple different levels. There's systemic regulation, including endocrine factors, which are hormones. Of course, there's nutrition, and also there's circulating inflammatory cytokines, which is usually associated with a person being sick. There's also local regulation inside the growth plate itself by these paracrine factors that are signal going back and forth between chondrocytes themselves. There's these extracellular matrix proteins that make up the cartilage tissue, as well as other intracellular mechanisms like signaling pathways, microRNAs, and epigenetic changes that goes on inside the cells. Now let's first take a look at the endocrine regulation of the growth plate. So when we think about hormone that regulate child bone growth, obviously the first one we think about would be growth hormone. So growth hormone is produced by the pituitary gland in the brain, which then goes to the liver to stimulate the production of another hormone called insulin-like growth factor, IGF-1. IGF-1 then goes to the growth plate to stimulate chondrocyte proliferation and hypertrophy, therefore speeding up bone growth. There's also evidence that the growth hormone can act directly on the growth plate that is independent of its effect on stimulating the production of IGF-1. And so in children with growth hormone deficiency, which are usually caused by problem in the pituitary that can't produce enough growth hormone, we usually see decreased growth hormone level, which then therefore leads to decreased IGF-1 level, and that contribute to the poor growth. The second condition that involves the growth hormone and IGF-1 axis is called growth hormone insensitivity, which is often caused by mutation of the growth hormone receptor. And so in children with growth hormone insensitivity, the growth hormone failed to activate its receptor in both the liver and in the growth plate, and therefore failed to stimulate the production of IGF-1. Now, normally IGF-1 provides a negative feedback that goes back to the pituitary to stimulate to limit the production of growth hormone at the pituitary. And so in growth hormone insensitivity, the low IGF-1 level actually leads to elevated growth hormone level. And over the years, geneticists have eventually found other growth conditions that also involve this growth hormone IGF-1 pathway. For example, similar to growth hormone receptor mutation, you could have a IGF-1 receptor mutation, and so that leads to growth hormone insensitivity and poor linear growth. Children could also develop IGF-1 deficiency, which has been found in children with mutation in a molecule called acid-late-bile subunit, or ALS. Normally, the ALS forms a complex with IGF-1 and with IGF-BP3 to stabilize the activity of IGF-1 in the blood. And so the loss of function mutation of ALS lead to destabilization of IGF-1 and therefore deficiency of IGF-1, and therefore that causes short stature. Anyway, in addition to the growth hormone and IGF-1 axis, there could be other hormones that could act directly on the growth plate to stimulate or inhibit growth. One example of that is glucocorticoid. So glucocorticoid could either come from the adrenal gland, which produces cortisol, which is a type of glucocorticoid. It could also come from exogenous medication, like prednisone, that are anti-inflammatory drugs, sometimes used to treat inflammatory disorders in children. So when glucocorticoid is used long-term or in high concentration, it could lead to suppression of bone growth in the growth plate. Thyroid hormone produced by the thyroid gland is another important hormone for child bone growth. So the thyroid hormone can act directly on the growth plate to stimulate growth. And also indirectly, the thyroid hormone can stimulate growth hormone production as well as IGF-1 activity. And so in children with hypothyroidism, we usually see low level of thyroid hormone, which is associated with low growth hormone level and low IGF-1 level, all of which contribute to their poor linear growth. And then during adolescence, estrogen and androgen produced by the gonads are responsible for the pubertal growth spurt. Both of these sex steroids can act directly on the growth plate to stimulate growth, but estrogen does so in large part by also stimulating growth hormone production that is happening during puberty. And lastly, there's a hormone that has been discovered relatively more recently called fibroblast growth factor, FGF21. It's produced mainly by the liver, and it was found to have a direct inhibitory effect on the growth plate. And as you are going to see next, FGF21 also plays an important role in the nutritional regulation of bone growth. Okay, so malnutrition in children often leads to poor bone growth, and it's normal to think that nutrition must play some important role in regulating all these hormones that regulate child bone growth, right? But before we get to that, let's first clarify what we're talking about when we use the word malnutrition. As we all know, normal linear growth requires the intake of adequate calorie, protein, vitamin, and other micronutrients. And I like to think malnutrition can be classified into two main types. One is called protein energy malnutrition, which is used to refer to a general nutritional deficiency or the fact that a child is simply not taking enough calorie, not eating enough food. Whereas the other type, it's micronutrient deficiency, which is used to describe the deficiency of a specific micronutrient like iron, zinc, iodine, and calcium. Now bear in mind, a lot of times when a child is not eating enough, you could have both things, but from a mechanistic point of view, it's better to keep them separate. So let's first take a look at what happened when when a child has protein energy malnutrition. So protein energy malnutrition slows linear growth in part by inducing a state of growth hormone insensitivity, which I've just talked about when it's a little bit like when a person has growth hormone receptor mutation. And so in a malnourished child, the growth hormone cannot efficiently stimulate IGF-1 production. And so IGF-1 level tend to be low. And again, because of this negative feedback, growth hormone level tend to be high. And we now know that part of this effect of malnutrition on the growth hormone and IGF-1 axis is mediated by FGF21. Because in both human and the mice, FGF21 level increases in the circulation after prolonged fasting. And it was found that this increased or elevated FGF21 level in the circulation decreases growth hormone sensitivity in both the liver and in the growth plate by downregulation of the growth hormone receptor at the mRNA level. There's also evidence suggesting that FGF21 directly interferes with some downstream growth hormone receptor signaling activity, which involves the JAD-STAT pathway. There's another effect of malnutrition on the growth hormone IGF-1 axis, and that is by suppressing the local activity or local action of IGF-1 signaling by increasing the level of IGF binding proteins. Now, IGF binding proteins is a class of protein or molecule that binds to IGF-1, and when IGF binding protein is bound to IGF-1, IGF-1 cannot act on the receptor to trigger IGF receptor signaling. And so in the local level, IGF binding protein negatively regulate IGF receptor signaling. And there's numerous reports showing that during fasting, local IGF binding protein level increases, and therefore that leads to suppression of local IGF signaling at the growth plate. There's another important effect of malnutrition on the endocrine system, and that is malnutrition can shut off the reproductive axis and therefore decreasing the level of estrogen and androgen, and therefore decreasing their effect on the pubertal growth spurt. There are evidence suggesting that this effect is mediated in part by another hormone called leptin that is produced by the adipose tissue. So the leptin level in the body more or less reflects the amount of body fat mass in a body, and it was found that a certain level of leptin is needed for the onset of puberty, and also a certain level of leptin is needed to keep the reproductive axis in an active state at any given time. So one could imagine in a malnourished child, because the overall body fat mass, it's going to be low, that tend to give low leptin level, and therefore that leads to shutting off of the reproductive axis, decreasing the level of estrogen and androgen, and therefore decreasing the pubertal growth spurt. So other than the growth hormone axis and the reproductive axis, malnutrition could also have other effects on other hormonal system, and that includes increasing the level of glucocorticoid because of more stress, and that's bad for growth plate. It also leads to decrease of thyroid hormone level, and again, that's not a good thing for the growth plate. All of these hormonal changes caused by protein energy malnutrition, therefore collectively impose a robust suppression of bone growth at the growth plate. Now, what about micronutrient deficiency? Back in 1996, Dr. Michael Golden proposed a classification of all kind of nutrients, which includes micronutrients, into type 1 and type 2, based on the way the body responds to its deficiency. The response to type 1 nutrient deficiency tend to be more specific, but the effect tend to be more gradual, whereas type 2 deficiency tend to lead to more general and more immediate response. In terms of global prevalence, the most common micronutrient deficiency exists for iron, folate, iodine, zinc, and vitamin A. But the mechanism by which the deficiency affects bone growth, or whether or not there's actually an effect specifically on bone growth, is not always very clear. And I'll try to talk about some of them here one by one. So for iron deficiency, we know that iron is needed for hemoglobin synthesis. And so iron deficiency commonly leads to anemia in children. And also in fetal life, iron is important for the brain development as well as overall body growth. And so iron deficiency during pregnancy is associated with low birth weight, but its mechanism in bone growth is not very clear. There's some evidence that iron deficiency leads to hypoxia, probably because of anemia, and that was known to inhibit chondrocyte proliferation. While there are others who found iron deficiency associated with decreased IGF-1 level as well as decreased IGF-1 activity, and that could also partly explain the poor growth associated with iron deficiency. Okay, now how about iodine and zinc deficiency? Iodine is needed primarily for the synthesis of thyroid hormone, and so iodine deficiency could lead to a spectrum of diseases affecting the thyroid gland, including goiter, hypothyroidism, and cretinism. Now, we've already talked about how important the thyroid hormone is for bone growth, so we've got that covered. For zinc deficiency, there's some interesting data found in rat that show zinc deficiency causes decrease of IGF-1 level, but I think in human, actually whether there's an effect of zinc deficiency on bone growth is not clear. In comparison, the mechanism for vitamin A deficiency in bone growth, it's a little bit more well elaborated, and so I'll go briefly into the mechanism here. So vitamin A deficiency typically could lead to blindness and also sickness in a child, and there's also some specific effect on bone growth, and so it could lead to linear growth failure. And there's some strong evidence that comes from genetic condition that lead to increased vitamin A level, and that causes accelerated bone maturation in children, as well as craniosynostosis, which is a condition to describe early closure of the scalp bone, which is another sign of advanced bone maturation. And we've also have some unpublished findings that vitamin A level actually positively correlate with height in young children in the blood. In terms of mechanism, the effect of vitamin A on bone growth is likely mediated through its metabolite, which is retinoic acid. Because retinoic acid receptor is expressed in the growth plate, and we know that deletion of retinoic acid receptor, particularly the gamma subunit, lead to shortened proliferative zone, decreased chondrocyte proliferation, decreased overall growth plate height, as well as stunted growth. And so one could imagine Vitamin A deficiency could similarly lead to decreased retinoic acid level, therefore decreased retinoic acid receptor signaling, and therefore could similarly lead to all these problems in the growth plate. Anyway, I just want to point out that there are different mechanisms that could involve for each of these micronutrient deficiency, and it's not very clear whether or not all the type 1 micronutrient would involve a certain class of mechanism, whereas type 2 involve a different kind of mechanism. And rather than spending more time talking about this, I'm going to move on and talk about another systemic factor that regulates bone growth, and that is circulating inflammatory cytokines, which probably contribute to the poor growth in children suffering from systemic inflammatory disorders like juvenile idiopathic arthritis and Crohn's disease. So it was found that these circulating inflammatory cytokines, including interleukin-1, 6, and tumor necrosis factor alpha, they can act directly on the growth plate to suppress growth. This effect has been demonstrated using an organ culture system where we can isolate fetal red metatarsal bone, and we can place them into the culture media, and they will continue to grow for a while because they contain the growth plate. And you can put different kind of things into the media and then see what it does to the growth plate. So for example, if you put IGF-1 into the media, it would lead to increased bone growth. And guess what happened when these inflammatory cytokines were added to the medium? It lead to robust suppression of bone growth in these bones, therefore suggesting there's a direct inhibitory effect on the growth plate. It's worth mentioning that some of these systemic inflammatory disorders could also inhibit linear growth through other mechanisms, that is, other than these inflammatory cytokines. For example, these disorders typically impair nutritional intake, and so that would contribute to the poor growth. Also, inflammatory disorders are often treated with glucocorticoid medication like prednisone and dexamethasone, which, as I mentioned before, could also lead to inhibition of bone growth. Now, of course, you can think of it from the other direction. Like in the developing country, poor nutrition could lead to increased episode of infection in childhood, and therefore lead to increased exposure of the growth plate to inflammatory cytokines, and therefore leading to growth suppression. But there's another interesting connection between nutrition, inflammation, infection, and growth, and that involves the microbiome, which I think perhaps deserves its own talk here. And so I'm not going to go into detail about the microbiome here. I would like to just point out a few things. So the human gut microbiome refers to a collection or aggregate of microbes that reside in our intestine. And we now know that the gut microbiome probably contribute to nutritional intake, and also different kind of nutrition could shape our microbiome into different forms. And so, for example, poor nutrition or poor nutritional status could lead to establishment of microbiome that is actually not good for you, so it could negatively affect your nutritional intake and also lead to increased infection and inflammation. And so that provides another possible mechanism by which malnutrition, especially in the developing world, could lead to child stunting, both affecting your infection and affecting your microbiome. Okay, so far all I've talked about involves systemic regulation of the growth plate, but let's not forget linear growth could also be regulated locally within the growth plate itself. For example, there are these paracrine signaling factors that go back and forth between chondrocytes themselves. There are cartilage extracellular matrix proteins like acrikin that also contribute to the regulation of growth, and also there are intracellular mechanisms such as signaling pathways and epigenetic changes. Now, genetic defects in any of these areas could potentially lead to linear growth failure. For example, mutation, actually activating mutation of the FGF receptor, FGFR3, causes achondroplasia, which is a rather common form of severe short stature found in children that is due to genetics. And mutation of the gene acrikin causes a spectrum of growth disorders that includes one of the most severe skeletal dysplasia. And I'm going to give you a few examples by which nutrition could affect or regulate some of these intracellular mechanisms that therefore affects growth. So the first one is nutrition. Remember that malnutrition or prolonged fasting increase the level of FGF21 in the circulation, and that leads to growth hormone insensitivity, therefore affecting bone growth. But in fact, FGF signaling is also a paracrine signaling pathway. And FGF signaling is a strong inhibitor of the growth plate because I just mentioned activating mutation of the FGF receptor gives you achondroplasia. And so one can imagine elevated FGF21 level that is caused by malnutrition could also act locally inside the growth plate because FGF21 could bind to and activate FGF receptor, FGFR3, and that lead to suppression of bone growth, a little bit like what happened in achondroplasia. Another effect of malnutrition on local mechanism involve the mTOR signaling pathway. Now, mTOR is the target of a molecule called rapamycin, which is a macrolide produced in bacteria. It was used clinically as an immunosuppressant to prevent organ rejection after kidney transplantation. We know that at the growth plate, treatment of rapamycin lead to suppression of mTOR signaling pathway, and that also lead to suppression of chondrocyte hypertrophy and suppression of bone growth. Now, where's the connection with nutrition? We know that in addition to rapamycin, another thing that would inhibit mTOR signaling pathway is malnutrition. Because malnutrition leads to decreased IGF signaling, and it turns out decreased IGF signaling is a signal that leads to decreased mTOR signaling in chondrocytes. Additionally, malnutrition induces a low energy status in all kinds of cells that includes chondrocytes, and that was found to be mediated through a molecule called AMPK. And it was found that this low energy status also, it's a signal for suppression of the mTOR signaling pathway. And so one of the local mechanisms by which malnutrition inhibits bone growth involves suppression of the mTOR signaling pathway in chondrocytes themselves. Okay, so I've talked about the physiology of child bone growth, and I've talked about how the growth rate is regulated in the growth plate. Now let's move on and talk about how the timing and duration of bone growth is also regulated. Normal linear growth ceases during adolescence, but interestingly, growth does not stop suddenly. In fact, you might find it interesting that linear growth begins to slow even before birth. If you just look at this graph, in mid-gestation, the human fetus grows in length at an enormous rate, which is more than 100 centimeters per year. But by birth, this rate has already fallen to about 50 centimeters per year. And by mid-childhood, this rate is way down to about 5 centimeters per year. There's a brief comeback, so to speak, during puberty, but then this decline continues until growth essentially stops. So the question is, what drives growth to slow with age? One possibility is that this deceleration might be due to some systemic mechanism, for example, change in hormonal level in the body. However, for example, the level of circulating IGF-1 and growth hormone do not decline in a way that could account for the decline in growth rate. If you look at the orange line in this graph here, in fact, IGF-1 level increases through childhood until mid to late adolescence. But more importantly, transplantation experiments show that when you take the growth plate of a young animal and put it into an older recipient, like this one, it will continue to grow at a high growth rate. That means depending on the age of the original growth plate rather than depending on the age of the recipient. And therefore that suggests that growth rate as well as the slowing of growth that happens with age, it's not due to a systemic mechanism, but rather to a local mechanism that is inside the growth plate itself. And we call this growth plate senescence. Based on the concept of growth plate senescence, child bone growth gradually slows with age because the growth plate, which is also the driver of bone growth, gradually gets older and older with age. That means the growth plate ages. And that's true because if you take the growth plate at different ages and take a look at it, you will see a lot of functional changes. As the growth plate ages, they don't proliferate, the chondrocytes do not proliferate as quickly. And also structurally, there's a decline in number of cells in each of these zones. The height of each zones becomes shorter. The chondrocytes do not expand as much as earlier, and the column become more widely spaced and so on. And near the end of growth plate senescence, when the growth potential in the growth plate is exhausted, at least in human, okay, the epiphysis will be fused, and linear bone growth will essentially stop at around 20 years of age. So in experimental animal, we can assess growth plate senescence easily because we can take out the growth plate and just take a look at, we can make sections of them and we can look at them under the microscope. But in children, of course, we can't do that. But what we can do is we can take X-ray images of their growth plate and determine the level of maturation. And we call this a child's bone age. It turns out the child's bone age is a fairly good radiological marker for growth plate senescence, which is why pediatric endocrinologists often use the bone age to predict a child's final adult height using the Bailey-Pinot table. So I'll give you an example. For example, if we look at a child's X-ray images and he's a boy, and if we found that his bone age is 13 years and 6 months, then we know that he's about 90% of his final adult height. In other words, we know that he has already used up about 90% of his growth potential in the growth plate. Another question that we have is, now growth plate senescence occurs with age or with time, but we wonder if growth plate senescence really is a function of time. That means there's a true biological clock in the growth plate and tell them when to stop growing, or might it be instead be a function of cell division? That means there might be some cell cycle counter in the chondrocyte, or that there's a certain proliferative capacity in these chondrocytes which gradually get exhausted as you get more growth or more proliferation of them. And that's what drives growth to slow and eventually ceases. To distinguish these two possibilities, one could try to inhibit growth and see whether or not then you would slow senescence. Because if senescence is a function of growth, then slowing growth would slow senescence. But if senescence is a function of time, no matter how you slow growth, that wouldn't affect the passage of time, and so you wouldn't slow senescence a bit. And in fact, we have used multiple models. We've done a tryptophan deficient model. We've tried to inject glucocorticoid in rabbits, and we've done a hypothyroidism model in rat. And all these models point to the fact that senescence is a function of growth, because prior inhibition of growth really slow growth plate senescence. So let me just repeat it one more time. So growth plate senescence is a function of growth rather than of time. Now this finding has an important implication, and I want you to remember this, and that is, while growth inhibiting condition might temporarily slow down growth, it actually might help preserve the growth potential in the growth plate, and so once these conditions get resolved, growth can be resumed. And this increase in growth velocity following a temporary growth delaying condition is also a clinical phenomenon that we call catch-up growth. So in the past, people have tried to understand the mechanism for catch-up growth, and people have proposed different theories. For example, there's the neuroendocrine model for catch-up growth that proposed the brain may be responsible for sensing the size of different organs and body parts, and so when it sees a size mismatch, it will try to produce more hormone to that organ and try to speed up growth a little bit. But after many years of research, there's not much evidence supporting this model. And instead, we now know that catch-up growth can be partly explained by delay in growth plate senescence, because growth inhibiting condition slows growth plate senescence and therefore preserve the growth potential in the growth plate. Once the condition is resolved, when you compare these two growth plates, this growth plate that has previously undergone growth inhibition now looks a little bit younger than this growth plate that is of the same age. And so this growth plate would tend to grow faster, and that's what you see. That's catch-up growth. Unfortunately, depending on the duration and type of growth inhibition, catch-up growth is often, more often than not, incomplete, leading to some permanent decrease in final height. And one area that is under active investigation is actually whether or not you could, or what kind of nutritional supplement you can use to help improve this incomplete catch-up growth in the recovery phase. There's some early study in animal try to elucidate the factors contributing to the completeness of catch-up growth. For example, people found that the type and severity of growth inhibition matters, the duration of growth inhibition matters, and interestingly, they also found that the stage of development by which the growth inhibition happens also affect the completeness of catch-up growth, such that the earlier the inhibition, the less complete it is, the catch-up growth. And perhaps that is why the first 1,000 days of life has gathered so much attention, maybe deservedly so, as a critical period of growth in a child. Of course, we know that this is a time when a lot of the brain development is happening, but maybe also for linear growth, because this is a time when linear growth is happening most rapidly, and so the most growth potential is at risk of permanently lost if any kind of growth inhibition occurs, leading to incomplete catch-up growth later in life. So one final point I would like to make about senescence is that in addition to growth that drive growth plate senescence, there's another thing that speeds up growth plate senescence. It's actually a hormone, and that's estrogen. We know that estrogen drives growth plate senescence thanks to some rare cases of patients who who either has estrogen receptor mutation or mutation of the aromatase, which is the enzyme responsible for estrogen biosynthesis. So in these patients, they either have decreased estrogen receptor activity, or they have decreased level of estrogen. And in this patient, bone growth happens until much later in life, which means they have delay or absence of growth plate senescence. The mechanism by which estrogen drives growth plate senescence involves depletion of the resting chondrocytes, the stem cell, during puberty. So we know that estrogen actually pushes these stem cells into the proliferative column during puberty, which is perhaps why growth stops pretty quickly after the completion of puberty. And so if you put this together, you might be thinking, well, malnutrition might have multiple mechanisms to delay growth plate senescence. So one is, for example, of course, malnutrition lead to slowing of growth, and that lead to slowing of growth plate senescence. And the second mechanism might be that because malnutrition also shut off the reproductive axis because the lack of leptin, and so that takes away the effect of estrogen in speeding up growth plate senescence during puberty. Now, unfortunately, depending on the timing of malnutrition and the recovery, this second mechanism may or may not benefit a child. For example, if malnutrition occurs during adolescence, yes, it's going to delay puberty, and so it's going to delay growth plate senescence. In terms of the final height, yes, it's going to benefit because delayed senescence gives you more time to grow, and so it's going to help. But if malnutrition is happening in early life, which is unfortunately the case in the developing countries, and if these conditions get subsequently corrected, so for example, if a child gets adopted from developing world into a developed country, there's evidence suggesting this actually leads to early puberty, therefore earlier growth plate senescence, and therefore further reduction of final adult height. And so we need to be cautious when thinking about the relationship between malnutrition, timing of puberty, and also bone growth. All right, so I think you'll be glad that I've come to the conclusion slides here. Today we have talked a lot about the growth plate. Just want to remind you, children grow taller because of a process called endochondral ossification. That means using cartilage as an intermediate to build bones. And this process takes place at the growth plate. In each growth plate, there's complex regulation by different systemic and local mechanisms. There's hormones, inflammatory cytokines, endocrine factors, and intracellular mechanisms. Importantly, nutrition plays an important role in modulating all these factors, including hormones, local mechanism. It might even have an effect on the microbiome. We have learned that longitudinal bone growth doesn't stop suddenly. It gradually slows with age, and it's driven by a process that is a mechanism that is intrinsic to the growth plate itself. We call it growth plate senescence. Growth plate senescence is driven by growth itself rather than by time, and so condition that slows growth also tend to slow senescence. And importantly, because malnutrition is also one of these growth inhibiting conditions, and so it would tend to slow senescence. But that's a good thing because it helps preserve future growth potential in a child, and that allows catch-up growth to occur once the malnutrition, or if the malnutrition gets resolved. And that's all I have for you today, and thanks for staying with me. If there's enough time, I can take a few questions. Like 3 minutes? Okay. Sorry, a little bit too long.
We started a little late.
Yes?
Thank you so much for your presentation and the level of detail you went into. I had one question about leptin. You had mentioned early on that increased leptin pushes bone growth during puberty or high levels of leptin?
You need some leptin it's more like a permissive signal for things to happen. So,
Oh, sorry.
I'm just saying, if you just give a person leptin, more leptin, it's not going to do anything. But if you take away leptin, it's going to do a lot of things.
Okay. Oh, thanks. Cool. I just want to ask, so leptin also, associated with high body fatness oftentimes, and you also said inflammatory cytokines decrease bone growth. Often we see in people with high body fatness, obese kids, we see less growth during puberty as well. So have you looked at, and more inflammation, so have you looked at like a sweet spot or relative abundance between these compounds? Like at what point do the levels of leptin and interleukin-6 and these other compounds result in less growth than stunting during puberty?
Okay, that's an interesting question. So what I know is obesity usually is associated with more, it leads to advanced bone maturation because kids of obese tend to start growing a little bit faster. That's what actually happens. But they end up probably being not taller. They might even be a little shorter over time. There could be complex regulation. One thing is if you're fat, your leptin get turned on a little bit earlier, and so you would have early puberty. Early puberty, that means your puberty growth spurt happens a little bit earlier than other people. So if you're the obese child in your class, you might start growing a little bit faster than some other people. But as I mentioned, if you have earlier puberty, you also advance your growth plate senescence. And so your growth plate gets fused a little bit earlier than other people, and so you stop growing a little bit earlier. Also, obesity tends to be associated with increased cytokines, and so that's an counter mechanism that would offset your increase in growth due to early puberty, which is associated with increased growth hormone level at the time. And so a child would be maybe a little bit taller in the class for a while, but he might end up being a little shorter than his genetic potential would allow. Doesn't mean he's going to be short, but he's going to be maybe a little bit shorter than he's allowed if he's not as fat. Yes?
Just two comments. One, I think there's a dimensionality to the complexity related to an inflammatory response and whether that inflammatory response might influence the nutrients available for stimulating some of these other processes. In other words, you've got a nutrition response, you have an inflammatory response, but you also have an interaction between those. And the second comment was related to other factors that will affect growth, particularly in adolescents and lower middle income countries, is early pregnancies or adolescent pregnancies, which in fact create a demand on the or change perhaps the way nutrients are partitioned either away from skeletal growth in a growing adolescent towards the developing fetus.
That's a very interesting comment. I mean, I haven't actually looked at this interaction, particularly the, pregnant adolescents, having resource switch away from your own growth. But I mean, this is something that would be interesting to look into.
This one's dead battery.
Oh, just have one. We can always continue the question in the.
Sunny, you're actually joining us for lunch. Yeah, okay, so I'm going to ask you to hold your question. So we can thank you very much for coming. This has been a fascinating talk.
All right.
And if you guys have extra questions, please stick around after. We still have a few places for lunch if you want to join us. Okay.
Sure. I'm going to report.